Recent scientific research has challenged long-standing assumptions about human aging, suggesting that our environment and lifestyle choices exert far greater influence over our longevity than previously understood. A landmark study published in Nature Medicine has provided compelling evidence that factors ranging from smoking habits to socioeconomic conditions play a decisive role in determining how quickly we age at a biological level, often overshadowing genetic predispositions by a significant margin. For generations, the scientific community has been captivated by the mystery of aging, pursuing various avenues of research to understand why some individuals maintain vitality well into their later years while others experience accelerated decline. Substantial resources have been directed toward genetic investigations, seeking to identify the hereditary components that might predetermine our aging trajectory. While these genetic studies have yielded valuable insights, they have failed to fully explain the vast differences in aging outcomes among individuals with similar genetic profiles. This groundbreaking research, drawing on health data from the UK Biobank's nearly 500,000 participants, approached the aging question from a different angle. Rather than focusing primarily on genetic factors, researchers examined 164 distinct lifestyle and environmental variables to determine their relationship with biological aging and mortality risk. This comprehensive approach allowed for a more nuanced understanding of how our daily choices and surroundings impact our health over time. The study employed sophisticated methodology centered around what researchers term a proteomic aging clock. Unlike chronological age, which simply tracks calendar years, this biological measurement analyzes proteins in the bloodstream that serve as molecular indicators of physiological aging processes. By examining these biomarkers, scientists can determine whether an individual is aging more quickly or slowly than their chronological age would suggest, providing a more accurate assessment of their true biological state. Through detailed analysis, the research team identified 25 factors significantly associated with accelerated aging and increased mortality risk. Remarkably, 23 of these factors are potentially modifiable, suggesting numerous opportunities for intervention. The identified factors span diverse aspects of human experience, including dietary patterns, physical attributes, socioeconomic indicators, lifestyle behaviors, and environmental conditions. While all 25 factors demonstrated meaningful associations with aging outcomes, 4 emerged as particularly influential: smoking status, socioeconomic position, physical activity levels, and living environment. Smoking showed associations with the largest number of diseases, while socioeconomic factors and fatigue frequency were close behind in their wide-ranging health impacts. Experts note that these findings underscore the importance of fundamental health practices over trendy wellness interventions. The research suggests that focusing on basic aspects of health that have been recognized for decades, such as avoiding tobacco, maintaining physical activity, ensuring adequate rest, and addressing social determinants of health, may yield the most significant benefits for longevity and disease prevention. Perhaps the most striking revelation from this research is the dramatic difference between environmental and genetic influences on mortality risk. The study attributed approximately 17% of the variation in death risk to environmental factors, compared to less than 2% explained by genetic predisposition for 22 major diseases. This tenfold difference challenges conventional wisdom about the primacy of genetics in determining lifespan. Researchers suggest that this finding should inspire optimism, as it indicates substantial opportunity for individuals and societies to influence aging trajectories through modifiable factors. Rather than viewing aging as predetermined by genetic inheritance, we can understand it as a process significantly shaped by environmental exposures and lifestyle choices, many of which remain within our control to change. The study also revealed interesting disease-specific patterns in the balance between environmental and genetic influences. Environmental exposures appeared particularly influential for conditions affecting the lungs, heart, and liver, organs often impacted by lifestyle choices and environmental exposures. Conversely, genetic factors maintained greater dominance for certain conditions, notably dementias and breast cancer, suggesting different diseases may have distinct environmental genetic influence profiles. Medical experts have responded enthusiastically to these findings, noting that they provide robust evidence for what many clinicians have long observed in practice: environmental factors and lifestyle behaviors profoundly impact health outcomes, often more significantly than genetic predispositions. They emphasize the substantial opportunity to reduce disease burden globally by addressing modifiable risk factors identified in the study. Health professionals also note that while the identified environmental factors explain a significant portion of mortality risk, they account for only 17% of the overall variation. This suggests many additional environmental influences remain to be discovered, potentially including chemical exposures and other factors not captured in the current analysis. Scientists in the field suggest several promising directions for future research. More detailed investigation into the specific biological mechanisms through which environmental factors accelerate aging could provide valuable insights for intervention development. Longitudinal studies examining whether targeted programs addressing key modifiable factors, such as smoking cessation initiatives, physical activity promotion, and socioeconomic support, can measurably slow biological aging would provide important validation of these findings. Researchers also recommend incorporating multiple types of biological data to develop a more comprehensive understanding of how environmental and genetic factors interact, potentially enabling more personalized approaches to preventive care. Additionally, replicating these findings across more diverse populations would ensure broader applicability of the conclusions. Despite its groundbreaking nature, the study has certain limitations worth acknowledging. The UK biobank population, while large and well characterized, may not fully represent global demographic diversity. Additionally, the observational study design cannot definitively establish causal relationships between identified factors and aging outcomes. Some associations may reflect bidirectional relationships or result from unmeasured confounding variables. Nevertheless, the implications for public health and individual wellness are profound. The findings suggest that public policies addressing key environmental factors, particularly socioeconomic disparities, tobacco use, physical inactivity, and housing conditions, could substantially improve population health and longevity. Similarly, individuals may find empowerment in recognizing that many factors influencing their aging trajectory remain within their personal control. Health experts emphasize that this research underscores the importance of prioritizing fundamental health practices that have demonstrated benefits over decades of research. While these basic principles of health maintenance may seem obvious, the study quantifies their impact with unprecedented precision, providing compelling evidence for their continued centrality in both personal health decisions and public health strategies. As our understanding of aging continues to evolve, this research represents a significant advance toward a more nuanced comprehension of the various factors influencing our biological aging processes. The findings point toward a future where aging interventions might be customized based not only on genetic profiles but also on comprehensive assessments of environmental exposures and lifestyle patterns. Such personalized approaches could help individuals identify their most significant modifiable risk factors and develop targeted strategies to address them. For societies facing the challenges of an aging population, these findings offer both reassurance and challenge. The reassurance stems from evidence that many factors influencing aging outcomes remain modifiable. The challenge lies in addressing deeply rooted social and economic inequalities that create disparate aging trajectories across population groups. The relationship between environmental factors and aging also raises important ethical considerations. If socioeconomic conditions significantly influence aging outcomes, ensuring equitable access to health-promoting environments becomes not merely a matter of individual choice but of social justice. This perspective shifts some responsibility for health outcomes from individuals to broader social structures and policies that shape the environments in which people live, work, and age. The study also carries implications for how we conceptualize aging itself. Rather than viewing aging as an inevitable, uniform process driven primarily by genetic programming, we might better understand it as a complex interplay between our biology and our environment, a dynamic process rather than a predetermined fate. This reconceptualization opens new possibilities for intervention and prevention strategies that could extend not just lifespan but health span, the portion of life spent in good health and functional independence. Educational initiatives about aging may need recalibration in light of these findings. Public health messaging typically emphasizes individual lifestyle choices, but this research suggests equal attention should be directed toward community-level factors and socioeconomic conditions that create the context for individual choices. comprehensive approaches addressing both individual behaviors and structural determinants of health may prove most effective in promoting healthy aging across populations. The technological implications are equally significant. If biological aging can be meaningfully influenced by environmental factors, technologies that monitor or modify these factors could become increasingly important in health maintenance. From wearable devices tracking physical activity to smart home technologies reducing environmental exposures, technological innovations may play an expanding role in creating environments conducive to healthy aging. As research continues to refine our understanding of how environmental factors influence aging, practical applications will likely emerge across multiple domains. Healthcare systems might implement more sophisticated screening for environmental risk factors, urban planners could design communities with healthy aging in mind, and policymakers might evaluate legislation through the lens of its potential impact on population-level aging outcomes. For a world increasingly concerned with extending not just lifespan but quality of life in later years, this research provides valuable guidance. By identifying the environmental and lifestyle factors that most significantly influence aging trajectories, it offers an unprecedented opportunity to reshape how we age as individuals and as societies. The path to healthier aging appears to lie not primarily in our genetic inheritance, but in the environments we create and the choices we make within them, a finding that simultaneously empowers and challenges us to reimagine the possibilities of later life.